Next, we'll move on to Chapter 21, which covers alpha carbon chemistry. Now, this is a modern sort of workforce in synthetic chemistry, and there's a lot of historically important reactions that we'll be discussing here. Before we get into the chemistry of it, we're going to talk a little bit about nomenclature conventions throughout the chapter. So if we have a compound like this with a ketone, an aldehyde, an ester, any other carbonyl group, we're going to be using Greek letters to note the position relative to the carbonyl group. Now the carbonyl group itself does not get a letter, only the positions next to it. So these two positions would be the alpha positions, the next two would be beta, the next one would be gamma, and so on and so forth down through the alphabet. Note here that this ketone is asymmetric and actually has two different alpha positions. This is going to be important later on. This can also show up in names for some of these derivatives. For instance, the neurotransmitter gamma-aminobutyric acid is a 4-carbon carboxylic acid with an amino group at the gamma position. This is especially relevant for a lot of biological or biochemical names that follow this naming convention fairly frequently. The reason why we care about this is that the alpha carbon of a ketone or an aldehyde or an ester or these other derivatives has a special reactivity to it due to keto-enol tautomerism. You might remember this from Chem 2131. We discussed briefly how a ketone is in equilibrium with its tautomer, which we call an enol. This is the same for aldehydes as well. We're also going to be talking a lot about the conjugate base of enols, which is called an enolate ion, so be sure you remember that name as well. Now, ketones and enols are in equilibrium, and as we saw in Chem 2131, this process is catalyzed either by trace amounts of acid or trace amounts of base. In fact, it's extremely difficult to stop this process from unless there are extraordinary experimental measures taken to suppress tautomerism. You can assume it's happening freely in solution no matter what the solvent. Generally, the carbonyl form is favored over the enol form. For example, if we have a compound like cyclohexanone, we have more than 99.99% of the keto form in solution and less than 0.01% of the enol form in solution. This is true for most of these derivatives, but there are some common examples that are exceptions to the rule, the most common of which being these beta-type dicarbonyl compounds. Now, in order for the enol form to be favored over the keto form, we generally have to have something happening that's making the enol form more stable in one way or another. In this case, about 10 to 30% is in the keto form in solution, while 70 to 90% is in the enol form. You might have already spotted what's going on here. For one, the enol form is conjugated, which adds stability to the structure, and for a second, there's an intramolecular hydrogen bond that can form, which also stabilizes this form of the molecule. Even though the enol tautomer is only present in small amounts in solution, it's quite reactive, and that's why we care about it so much. So if we have a compound like acetone, even though a tiny fraction of acetone is present as the enol, this enol has a resonance form which puts a negative charge on that carbon. So this is what determines the reactivity of these types of species and makes the alpha position of ketones, aldehydes, and other compounds fairly nucleophilic. Generally speaking, enolates are going to be more useful synthetically than enols since they're more reactive. Enolates can be formed from carbonyl compounds by treatment with an appropriate base. If you treat a carbonyl compound with a base, you pull off this proton, leading to a negative charge which is resonance stabilized and therefore quite reactive. You'll often hear enols referred to as bidentate nucleophiles. Bidentate comes from Latin meaning two tooths, because both the oxygen and the carbon have nucleophilic character and could potentially react with electrophiles. So it is possible to have reactions which either react with the carbon or with the oxygen. So for example, if we treat this compound with an electrophile, it could react at the oxygen to give us this sort of ether derivative. This is less common, and we won't actually see any examples of this in this chapter. Instead, the vast majority of reactions will happen due to C addition, where the carbon acts as the nucleophile. So it's worth noting here how I've drawn the arrows for the carbon addition. The reason why I've done this is it's convention to show the most stable resonance form of a molecule as being the one that reacts. So I'm going to show this molecule in the oxygen anion form, and I'll have that add to the electrophile as a nucleophile. Occasionally in the book you will see the carbon form used directly to react with the electrophile. There are some reactions where this will shorten our mechanistic path a little bit and reduce the number of arrows that we'll have to draw. This is also fine, although I'll try to use the most stable resonance form convention throughout. So how do we make these enolates? What kind of base do we choose? Well, as we've seen before, this all depends on the pKa values of the proton that we're trying to remove. Here are three representative compounds: acetone, acetophenone, and acetaldehyde. I've shown the most acidic proton on all three of these explicitly. The pKa values are 19.2 for acetone, 18.3 for acetophenone, and 16.7 for acetaldehyde. So this is a pretty familiar range. Generally speaking, most carbonyl compounds have a pKa which is close to the range of alcohols. So what this means is that a very common base for forming an enolate is going to be an alkoxide, the conjugate base of an alcohol. If we choose this as a base, we can remove this proton to generate the resonance stabilized enolate and form the alcohol as our conjugate acid. There are a few things to remember about this, which is that proton transfer is fast and reversible. Generally speaking, the enolate form is going to be present at a lower concentration, although this does depend some on the relative pKa values of the bases that we're going to use. In this case, if we use an alkoxide, ethanol has a pKa very close to 16 to 17, which means that at equilibrium we will favor the protonated form of the carbonyl compound rather than the enolate. It is possible to form enolates quantitatively and irreversibly by choosing a suitably strong base. For example, sodium hydride can deprotonate a carbonyl compound to generate the enolate quantitatively. Not only is sodium hydride a strong base, but hydrogen bubbles out of the solution after this reaction is done, making this effectively irreversible. LDA is also a very common base for the generation of an enolate which is irreversible. Now I'd like to point out in this case that I am deprotonating an aldehyde. The aldehyde proton is not appreciably acidic and we will never remove it with a base. Instead, we're going to be generating the enolate of the aldehyde plus diisopropylamine as our conjugate acid. In this case, the proton of our conjugate acid is 36, which is well above the expected pKa range of the alpha protons, meaning that this reaction is irreversible. Because the pKa difference is far too high, this reaction will lie entirely over to the right. Just as a reminder, equilibrium constants for these reactions can be estimated with 10 to the delta pKa between the acid and the conjugate acid. So in this case, there's about 20 pKa units separating our alpha proton and our diisopropylamine, meaning the equilibrium constant for this reaction should be on the order of about 10 to the 20. This is effectively irreversible. So let's take a look at another one to just test our knowledge on this. Is this reaction that I'm showing here between a beta dicarbonyl compound and sodium hydroxide reversible or irreversible? I'll give you the pK for this. The proton that's between these two carbonyl groups has a pK of about 9. Draw the reaction that would occur, including all products and all resonance forms of the conjugate base, and decide if this reaction ought to be reversible or irreversible. Unpause the video when you're ready to see the answer. So this reaction would produce this anion as its conjugate base, which is resonance stabilized. In fact, for this we can actually draw 3 good resonance forms, 2 of which have the negative charge on an oxygen atom. The conjugate acid that we get in this case is water, which has a pK of approximately 16. Since these two pKa's are separated by quite a large amount, this reaction is also irreversible. So if we need to deprotonate an alpha-beta dicarbonyl compound like this, we don't need a strong base like LDA. Sodium hydroxide will do this irreversibly. Again, if we wanted to estimate the equilibrium constant for this reaction, we could take 10 to the delta pKa and get an equilibrium constant of approximately 10 to the 7th. So let's take a look at an example of why we would want to do this. One reaction that we can do with enolates is alkylation at the alpha carbon, forming a new carbon-carbon bond. In this case, I'm taking cyclohexanone, forming the enolate with LDA, and then adding a good electrophile, Rx, to form a new carbon-carbon bond. This reaction proceeds in 2 stages. First is irreversible enolate formation with LDA to make the enolate, which will act as our nucleophile. Then I'm going to treat it with an electrophilet carbon, which will react at the C position to form a new alkylated carbonyl derivative. Now, one thing that we need to watch here is that this step is an SN2 substitution, so the usual restrictions about SN2 apply. This is best with a methyl or primary electrophile. We don't want too much branching, etc. Now, you might be wondering, why didn't I use ethoxide to deprotonate this and make the enolate in this case? Well, in this reaction, there'd be too many bases or nucleophiles present if I added ethoxide. Remember that with ethoxide, the equilibrium actually does not favor the formation of the enolate. So if I treat cyclohexanone with sodium ethoxide, I'm going to have a significant amount of sodium ethoxide in solution when I add my alkyl halide. Sodium ethoxide is also a very good nucleophile, so the likelihood of me getting the product that I want as the major product is fairly low. This is a little more complicated if we have asymmetric ketones. So if I take a ketone like this, methyl cyclohexanone, I have two different alpha positions, which means that I can form two different enolates. Now, if you look at the structures of these enolates, you might notice that one enolate is predicted to be more stable than the other. The top enolate, shown with the blue arrow, we're going to call the thermodynamic enolate, which is the enolate which is more stable. Generally speaking, the more substituted an enolate is, the more stable it's going to be. The other one we're going to call the kinetic enolate because it is formed by the removal of the least hindered set of protons. So as we've seen before, if we have a case where we have two different alpha positions, we can control which position gets deprotonated by changing the conditions under which we do the deprotonation. For the kinetic enolate, it can be favored by using a very bulky base at a low temperature to suppress equilibration. For the thermodynamic enolate, we can favor it by treating it at higher temperature, which will allow equilibration while we remove the proton. So here's some practice. Draw the missing pieces for these reactions. So in this first process, I'm taking this ketone here and I'm treating it with a bulky base at a low temperature. Then I'm treating the enolate that I form with butyl iodide in order to alkylate the alpha position. Which proton do I remove here and which product do I get? How would this change if I used a different base and I did this reaction at a higher temperature? Which enolate would I alkylate in this case? And then finally, if I want to place two different alkyl groups on the two different positions of this ketone, what sequence would I need to add my base and my electrophile in order to get these two products? Take a few minutes to work through these and then we'll compare notes in a moment. For the first reaction, treatment with LDA at a low temperature would tend to form the kinetic or less stable enolate. Here we've removed the more accessible primary proton versus the less accessible tertiary proton. If I treated this with butyl iodide, I would alkylate specifically at that position, giving me this as the major product. If I treat the cyclic ketone with sodium hydride at a higher temperature, this will favor the formation of the thermodynamic or more stable enolate. Therefore, when I add my alkyl halide, I'm going to favor reaction at that position. For the third sequence, for the first reaction, it actually doesn't matter so much which base I use because this ketone is symmetrical. So I would need to choose any base sufficient to generate an enolate quantitatively, followed by treatment with the appropriate alkyl halide. For the second reaction, it does matter which base I use. Since I'm trying to alkylate the least hindered position here, the secondary carbon over the tertiary carbon, I would choose a bulky base at a low temperature, followed by my electrophile. Next, we'll talk about a related reaction which will allow us to add a halogen to the alpha position of ketones, aldehydes, and carboxylic acids.